So I will probably give you some basic concepts that all of you have. I mean, you all know that eukaryotes have, keep their DNA in the nucleus well preserved in structures called chromosomes. And those chromosome structures are characterized by thin chromatin fibers. And the basic unit of chromatin fibers is a structure called nucleosome, which consists basically of a core made of proteins. Here are listed the 4 histone proteins that form a tetramer and a dimer, and together they form an octamer, over which about 150 base pair of DNA wraps around it. And this unit is repeated every 200 base pair in our chromatin on average. And this packing of our DNA is extremely useful for keeping a very long DNA molecule in a small volume of our nuclei. But in a way, biochemically, those DNA-protein interaction prevents the ability of other factors that are involved in transcription, replication, repair, or recombination, namely all the factors that need to handle and to interact with DNA to productively interact with site-specific recognition sites. So there is a problem for the cell. If there is a factor that needs to bind the DNA, how this process is carried out. I mean, we all know from our textbooks that chromatin it's bulkily divided into heterochromatin, which consists of chromatin that it's closed, where those nucleosomes are nicely packed together, and chromatin that it's more open, where those nucleosomes tend to be more spaced apart. And basically, we now know that closed chromatin and open chromatin is the result of a combination of complex reactions that we tend to call epigenomic marks that are put by our cells. And one of the most conserved mechanisms by tagging nucleosomes and by making those nucleosomes more open or closed, it's called covalent modification of the N or C terminal tails of those histone proteins. So basically there are enzymatic activities. Here are listed some of the most common, like histone acetyltransferases, kinases, histone methylases, and of course the enzyme that do the reverse reaction that use the N-terminal tails of histones as main target for the deposition of those marks. And of course, you can imagine that those histone tails are decorated with all of these marks, pretty much like our desk is in our office. It's decorated with post-it, where we have a memo of what to do or what we have to do in the next days or in the same day. And so you have to imagine that combination of these modifications are spread and are decorate our genome. And so basically, although every cell in our body contains the same DNA filament and the same sequence, basically different cells in our body are decorated with different histone tags, and that makes our cells more accessible in certain regions of their genome for certain factors and in other cells for others. But this is just one of the mechanisms by which the cell creates accessibility to DNA. We do also have another family of factors that are called chromatin remodelers, and those chromatin remodelers do not make any covalent modification. They just use the energy present in this molecule, the ATP, and they use the energy for pushing nucleosomes or for evicting nucleosome, removing the octamer, or just exchanging histones with other histones or histone variants, or by extruding the DNA from the nucleosome, for example, to facilitate reaction like DNA methylation. And of course, in recent years, it is obvious that probably we have to rewrite our molecular biology books, because the ancient concept of a gene with a promoter, exon, intron, and one main transcript that starts at the TSS and then and it's polyadenylated is probably going to be replaced with this view where basically our genome is pervasively transcribed, and it's probably a long continuum of transcripts that go in both directions. And this will not be the topic of my talk, but we'll probably hear in the following days some interesting talks where non-coding RNA plays an important role in gene expression and chromatin organization and packaging. So basically, the main idea that I hope I have delivered in this first 10-15 minutes of my talk is that we probably have one genome, but many epigenomes. And the many epigenomes are the result of the modification that proteins associated with the DNA have and that are established during the differentiation, the development of our body. So as we have genetic defects that result in loss of protein production or, I don't know, truncated protein, we also have epigenetic defects where basically the same sequence in two different organisms or in two different cells or in the same tissue are decorated with certain modification, and then, you know, this domain is active, it's fully transcribed, that then at some point, if something happens, this DNA, it's packed and it's not anymore available for transcription, and that may cause, of course, an epigenetic alteration in principle. The biological question that my lab tried to focus on a couple of years ago was to understand how nucleosome remodeling reactions, namely all the reactions that are catalyzed by those multi-subunit complex that use the ATP to push and slide nucleosomes, how these reactions in the context of covalent modification interact with this and make a genetic and biochemical network. Basically, how chromatin remodeling reactions are regulated by covalent modifiers of chromatin or other chromatin factors like even non-coding RNA that might be involved in the regulation. And so we are basically interested in how the cell receives signals in order to integrate those signals into complex regulation of a specific genomic stretch. So basically, we don't know how, you know, once we get signals, we can have something active, and then, you know, upon other signals, that is repressed. And this is probably a complex combination and crosstalk occurring between chromatin remodelers and covalent modifiers and other histone proteins. And the fruit fly is a great system because to study this type of this biological problem, because you can do genetics, and there are well-established tools to do genetics. You can squash ovaries or embryos or adult flies, and you can extract their proteins, so you can do decent biochemistry. Plus, they have a very potent tool because some of their cells have a gigantic interface chromosomes where you can basically look in vivo what's going on in those chromosomes. You can study the structure, the activation of some of the genes. So the chromatin remodeling complex that we use as a model factor to study how remodeling reactions are regulated is called ISWI, and it's highly conserved along the evolution. It's this green protein, and very quickly what ISWI does, basically it binds to nucleosomes, it uses the energy of ATP to push nucleosome and pack nucleosome in a more close packed structure. And in fact, indeed, in vivo, if you look at those chromosomes, those are salivary glands polyethylene chromosomes. Those are thousands and thousands of chromatin fibers that are aligned in a cell that has, you know, never divides, and it's always resting in interphase. So those are not mitotic chromosomes. Those are interphase active transcribing chromosomes. And if you lose this I-switch activity, so this chromatin remodeling activity, what you observe is a massive chromosome organization alteration. So basically, those chromosomes tend to puff, tend to disorganize themselves, which is consistent with the biochemical activity we found in vivo. I-switch normally in vivo tend to pack nucleosomes. So if you lose it in vivo, then chromatin tends to become looser. And if you do microarray studies, what you observe is that if you lose I-switch, many genes, you know, are de-repressed. Again, very consistent with a possible role of I-switch in packing chromatin, keeping it close, and maintaining, you know, transcriptional repression in many domains. And so what we were started, you know, to do was to, you know, classify chromatin IP. I'm sure that today we will hear a lot of work presented where this technique is shown. So basically, you cross-link the DNA, genomic DNA, with proteins, and then you just cut proteins by physical means or biochemical means, and then you use an antibody, pretty much like when you do an immunoprecipitation, and you pull down those DNA fragments, and then there are different ways to identify what these fragments are, and we did it by using microarray that was customized for us. And basically, we looked at where I-switch binds in the genome, and what we found is that I-switch, to our surprise, it's not spread all over the genome, but tends to concentrate close to gene transcriptional start site, and in particular, a little bit after the transcriptional start site. And what is interesting is that although we have nice peaks for I-switch in many genes, there are, of course, in this case, there are two genes firing in two opposite directions, and we find I-switch here and there. But there are, of course, exceptions. We find also I-switch in introns, and we don't know whether those introns are transcribed or not. We find it in the middle of the gene, and of course, there are some exceptions. And although we knew that I-switch was binding near the transcriptional start site, we didn't know whether I-switch activity was concentrated and was acting at the transcriptional start site. And I remember you that I-switch basically moves nucleosomes. So we basically adapted a protocol that was developed in yeast to look genome-wide at nucleosome positioning in flies wild type or in flies that lacked I-switch activity. So basically, since there is an enzyme that degrades and digests linker DNA between two nucleosomes, if you digest chromatin with MNase, this mycococcal nuclease enzyme, you protect nucleosomal DNA. And if you extract this nucleosomal DNA and then you hybridize it on a microarray, then you can identify nucleosomes. And where there are peaks, of course, there are well-positioned nucleosomes. If there is a broad peak, then probably this is a position where nucleosomes tend to have a high mobile feature. So what we found is that near the TSS in wild type, we have a classic region that it's NFR. It's the nucleosome-free region, where nucleosomes are absent to facilitate RNA polyol enzyme to transcribe. And there are a couple of highly mobile nucleosomes before the TSS, but two very well-positioned nucleosomes after the TSS in wild type. If we lose I-switch activity, what happens is that basically what's before the TSS remains the same, but those two nucleosomes after the TSS become highly delocalized and highly mobile. And this is exactly the region where I-switch binds. So our conclusion was that the way I-switch regulates and represses transcription is by keeping those nucleosomes well positioned after TSS. This has been the work of a PhD student in my lab, Maria Toto, and Anna Sala, a postdoc, one of the first postdocs that I hired in my lab. To summarize, I-switch is a nucleosome-stimulated ATPase. It spaces nucleosomes. It organizes chromosomes. This has a role in gene expression because when you lose it, there is transcription that goes amok, and I-switch does this by regulating nucleosome around the TSS. And work done in other labs have clearly shown that I-switch is also involved in many other processes like DNA replication, epigenetic resecting after nuclear transfer, stem cell self-renewal, and morphogenesis of sensory neurons, and also regulation of the RAS pathway. So how is it possible that one protein that basically pushes nucleosomes around, that's what I-switch does, plays a role in so many reactions? And our hypothesis is that I-switch is probably a highly regulated protein. We know that certain acetyl group, those are the little tags deposited by covalent modifiers, can repress I-switch activity. We know that other known subunits associated with I-switch can regulate its activity. We know that there are specific post-translational modifications of I-switch that regulate its activity, and we don't know about other factors, other proteins, or who knows, non-coding RNA or other covalent modifiers. So since we were not sure that biochemical studies were successful in identifying all I-switch regulatory pathway, we developed an in vivo assay to identify, probably by saturation, through a genetic screen, all the factors that could interact with I-switch and regulate its activity. So this is a developing eye tissue, and those are the cells, and the chromosomes present in those cells, and this is the fly eye. And if we lose I-switch in those developing eyes, we disrupt pretty much chromosome architecture, and we have an eye which is smaller than wild type, it's rough, and has a phenotype. So we decided to use this phenotype, this disrupted eye, to screen, to cross it with mutagenized flies in order to look in the progeny for flies that had either a more disrupted eye or a recovered eye. In genetics, basically, this corresponds to enhancers of a phenotype or suppressors of a specific phenotype. And the rationale beyond this screen was that an enhancer should have been a factor that, when lost, probably in wild-type cells cooperates and work in concert with iSwitch, while the suppressor is an antagonist of iSwitch activity. So we did many screens and we isolated, several factors. And one, I would like to summarize very briefly, some of the stuff that we found. So we isolated SYN3A and RPD3. This is a highly conserved complex in mammalian cells also. This corresponds RPD3 to the HDAC1. It's a histone deacetylase. And what we found, to cut the long story short, is that iSwitch normally is inhibited by specific acetylation of chromatin. And if iSwitch now wants to productively interact with that chromatin, it recruits the SYN3A RPD3 complex, it pulls it into the site that has to remodel, the deacetylase that chromatin, and now iSwitch can remodel and push this nucleosome. This has been the work of Josalba Burjo, a PhD student in my lab. Another factor that we identified in my screen was the PARP. PARP is the poly-ADP ribose polymerase. It's an enzyme that deposits ADP ribose moieties in proteins, and this has been found to be extremely important in the DNA damage response and also in the transcriptional activation. And basically, if I switch pack nucleosomes, and by packing nucleosome, it closes chromatin, and now this stretch of chromatin needs to be activated, PARP comes, among other factors, poly-ADP ribosylate iSwitch, and iSwitch detaches from chromatin, and then this chromatin domain can become transcriptionally competent. And this was the first identification of the poly-ADP ribosylation of a chromatin remodeling enzyme, and has been the work of the extensive work of Anna Sala, a postdoc in my lab. So the first take-home message is that chromatin remodelers are regulated by a complex network of covalent modifiers, and we think we have identified a few that escaped previous biochemical analysis. But we also isolated many suppressors, and one of the suppressors is called HSR-omega. And HSR-omega basically completely suppresses all these, chromosome condensation defects or eye sides, and phenotypic phenotypes caused by loss of iSwitch. So if you lose iSwitch and HSR-omega in the same eye, you recover the eye, you recover the ommatidia, you recover the chromosomes. And what is HSR-omega? HSR-omega is a non-coding RNA. So it's a locus in flies that encodes for a non-coding RNA associated with many HNRNP. And those HNRNP, together with HSR-omega, they form the so-called omega speckles. And the omega speckles are nucleoplasm compartments, shown here in green, that are devoted in the processing of mRNA molecules. So basically, they tend to process, like to cap them, to help the polyadenylation, to help their export. And they never map chromosomes or chromatin, shown here in blue, but they are always at the edge of chromosomes and often and almost exclusively mapping the nucleoplasm. And during a heat shock response, those nuclear speckles tend to cluster to, in a way, resist to the heat shock insult, so that those factors are preserved in specific compartments, so that at the end of the heat shock insult, then mRNA processing can start over again, without any interruption for the cell. And what we found is that when you lose iSwitch activity, basically in iSwitch mutant cells, those speckles lose their classic, classic wild-type distribution organizations. So instead of being scattered all over the nucleoplasm, they tend to form these trails, like if iSwitch activity was involved in the HSR-omega maturation. And in vivo, we found that indeed HSR-omega in green, that is in the nucleoplasm, I remind you, iSwitch that only binds chromatin on the DNA tend to touch each other. And they tend to, I wouldn't say co-localize, but because this is a very dynamic interaction, but they touch each other with a good correlation coefficient. So if we now do some biochemistry, if we try to pull down iSwitch and look whether in vitro also this RNA interact with iSwitch, what we clearly found is that you can easily IP iSwitch, and together with iSwitch, we can find HSR-omega. So we have here the first in vitro proof of an interaction between a chromatin remodeler and a non-coding RNA. And in vitro, iSwitch indeed can bind this RNA, non-coding RNA, by classic gel shift assay. And this non-coding RNA is able to stimulate iSwitch ATPase activity. So basically, iSwitch is not only stimulated by a nucleosome or by DNA, but also by the RNA. And the model that we came up with, and I don't have time to show all the data of this work, is that ISWITCH normally interacts with nucleosome to pack them, and that causes, of course, normal chromosome conformation. But what we found is that ISWITCH probably plays a non-canonical, previously not identified role in organizing omega speckles, because if we lose ISWITCH activity, we not only use chromosome condensation, but we also lose nuclear speckle organization. And that explains also the suppression phenotype that we have. And this is the work conducted by Maria Cristina Onorati, a postdoc in my lab. And what is interesting about this work is that there is a human homolog of this HSR omega that is involved in a neurodegenerative disease. Just take-home message, ISWITCH probably and chromatin remodelers not only interact with chromatin, but also with non-coding RNA, and probably they play an important role in organizing nuclear compartments. Just 2 slides to show you that we found that loss of ISWITCH has a profound effect in a model tissue that it's used for studying staminality, which is the Drosophila ovary. So basically, if we lose ISWITCH in those ovary, only 1% of female germ stem cells survive, implying that ISWITCH is an important factor in germ stem cell self-renewal, and we found this also in male gonads. And this is the work that's been going on with Maria Toto. And another phenomenon highly related to regeneration in mammals in flies called transdetermination, that it's an ideal system to study epigenetic memory. So basically, normally the wing tissue would develop into a wing in the adult tissue, an eye tissue would develop into an eye in the adult, but sometimes there are exceptions to this rule, and an eye can express some wing markers and vice versa when you do transplant. And what we found is that this is an eye tissue that should develop into an eye, but in green you see markers that are typical of the wing, and this process is called transdetermination, and the rate of transdetermination it's greatly decreased when you lose ISWITCH activity. So here we have probably the first example of a role of a chromatin remodeler in the regeneration of tissues in flies, and probably, who knows, this process is also conserved in mammalians, and this is some unpublished work. This is the project of Julia, a new PhD student in the lab. So basically, we would like to use all these ISWITCH regulators we found in order to look which one are involved in staminality, which one are involved in epigenetic memory, and then once we identify these factors, we hope we can recapitulate in vitro what is the mechanism of the interaction. So I think that's over. That's Mondello Bay in Palermo, and I would like to thank all the sponsors of our work and all the people in the lab, and of course, last but not least, let's hope for the best for Sunday. Thank you for your attention.